So, welcome back. In this segment, we are going to be talking about cell transport. And so last time we talked a little bit about the structure of the cellular membrane. And so we talked about the phospholipid bilayer as well as the proteins that are embedded within that phospholipid bilayer. Today, well, I mean, for me, it's the same day, but for you guys, it should be a different day. I just talked about cell membranes like 20 minutes ago. But for this lesson, we're going to look at how those structures impact how molecules move across the membrane. And so we're talking about things going in and out of the cell. And so one of the major determining factors about whether or not a molecule can cross this membrane is the properties of the phospholipids themselves. And so this membrane is selectively permeable. And permeable means that things can cross, right? So if something is entirely permeable, everything can come in and out. If something is impermeable, nothing can come in, nothing can come out. The membrane is selectively permeable, meaning some things can come in and some things can come out, but other things are trapped. And so we're going to look at how the phospholipids regulate what can go in and what can go out, and how the proteins alter what can go in and what can go out of this cell. We're also going to look at osmosis and how water potential impacts the way that things move. And not everything is permeable, but water is. And so we'll look at how the balance of things is altered when one substance is permeable and the other is not. Okay? So that's what we're about to talk about now. So we're going to start our conversation about cell transport on a topic that you should already be familiar with from Biology 1, and that's going to be the general principles of diffusion and passive transport. So diffusion is the tendency of molecules to move from a high concentration to a low concentration. So if I have a lot of apple bananas over here and very few apple bananas over here, the apple bananas are going to disperse randomly, and it's going to reach an equilibrium where eventually I'm going to have equal amounts of apple bananas everywhere because they moved from the high to the low concentrations. Okay? And now the molecules do not actively know that they're moving from high to low. They don't do it intentionally, of course, because they're molecules and they don't have intent. So molecules are moving randomly in every direction. Right? And so if I have a molecule up here, it can move in any direction, right? But the chances of any of these molecules moving this way, right, is overall higher, because there's so many of them, than any of the molecules down here moving that way, right? And so this one, if there's one molecule down here, it could go up, it could go that way, it could go that way, it could go that way, 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 right? And so all those 360 degree, or even more, I mean, there's really an infinite direction. Let's call it 360 degrees because that's easy to imagine. Right? It could end up going in that one direction that's up this way, right? But it's a low probability of that happening. Whereas these 360 molecules, right, there's a good chance that some of them are going to come down this way. Right? And so through this random movement and probability, molecules from a high concentration end up going to the low concentration area. There's nothing stopping the low concentration from going back up, but just the rules of probability make it not happen. Okay. So diffusion is not an intentional act by the molecules. It just so happens that when you have a bunch of molecules in one place and they're flying around in multiple directions, they're just more likely to bounce and hit and end up in a lower concentration area. Okay. So molecules will move from a high concentration to a low. This is true across a membrane as well, as long as the molecule can transport itself. As long as the molecule can go across the membrane, it will follow this diffusion pattern. So in this model here, I have three molecules up at the top. I have a triangle, I have a little dot, and I have a big circle. Okay. So these three molecules, right, are obviously in higher concentration up here than they are down here. I have no molecules down here. Okay. So through diffusion, the chances of them moving are down this way. Okay. So we got to look at the properties of the membrane to see whether or not these molecules can pass through the membrane. Okay. And so if we're looking at the small circle, the small circle is the same on any side, right? I take a circle, I spin it around, I flop it around. There's no difference from one side to the next. Okay, so this small circle I'm using to represent a non-polar molecule. There's no difference from one side to the next. And so if I look at my membrane, I have this large section of non-polar molecules. And now non-polar molecules like non-polar molecules. Non-polar molecules dislike polar molecules. So this tiny little dot is trying to move down. Okay. And so the first thing we have to ask ourselves, is the tiny little molecule small enough to fit between these phospholipids? And yes, it is, right? This gap between phospholipids is bigger than the molecule is. And so it can fit through. Right? Then we have to ask ourselves, is there anything that's going to repel this molecule from getting through this area? Okay. So this tiny molecule is nonpolar, it squeezes in this gap, it hits the nonpolar region. Okay. But because nonpolar things like nonpolar things, this little nonpolar molecule can pass right through. Okay. So small nonpolar molecules can move directly across the membrane along the concentration gradient. Let's look at our tiny triangle. Okay. So I have a small triangle. Right? And now one side of the triangle is not the same as the other side of the triangle, right? So the bottom's wide and the top is skinny. I'm using this to represent a polar molecule. And so this large non-polar region does not like this polar molecule. So we ask ourselves again, is this small enough to pass through the membrane? Right? And the answer is yes. This triangle is smaller than this gap. So this triangle can fit in the gap. And so it gets in, but then this region is like, yo, we don't like you. And so it bounces back out. Yeah. Now, we know that small polar molecules cannot pass through. Okay. That brings us to our last molecule. It's a circle, again representing non-polar. Okay, so the circle's the same on all sides. However, this circle's quite a bit bigger. We ask ourselves, can this circle fit through this gap? And the answer is no. Right? The gap is smaller than the circle is. Right? So even if it's non-polar, it still doesn't pass through. Okay. So this is the reason that only small, non-polar molecules can pass directly through the membrane. So if I have a small, non-polar molecule, it will move right through the membrane from a high concentration to a low concentration. And that is simple diffusion. So, not every molecule that our cell needs is small and nonpolar. Our cells need a way to move these polar or large or ionic molecules in and out of itself. And that's where protein channels come into play. And so when I have a high concentration on one side and I need the molecules on the other side, my cell relies upon a process called facilitated diffusion. Facilitate means to help, and in this case, the diffusion is being helped by the protein channel. And now these can be either very specialized protein channels and only moving one substance, or these can be very general protein channels and maybe they move every positive ion. So depending on the cell type and what kind of stuff it needs, it's going to have different protein channels associated with its membrane. In this case, I am moving a large polar molecule. And you can see I've drawn this protein to be shaped in a way that is complementary to triangles, because my molecule that I've drawn is a triangle. This is to indicate that the shape of the protein matches or in some way is complementary to the shape of the molecule that it moves. And now protein shapes and molecule shapes are far more convoluted than triangles. But it's a model, and that's how models work. They're symbolic, simple representations of complex ideas. And so this triangle has a high concentration up here, a low concentration down here. Once it finds the appropriate protein channel, it will move from high to low concentration. So it's still a passive transport. No energy is required to do this, but a protein channel is necessary because this molecule's qualities make it so it cannot pass through the membrane directly. Cool. So those are our types of passive diffusion, passive transport that did not require any energy exertion by the cell. Those things moved via diffusion from a high concentration to a low concentration, as is a natural tendency of all molecules to do. However, there are circumstances where cells need to be able to move molecules against the concentration gradient. Now, whether that be to dump waste out of the cell or to take nutrients into the cell needs to be able to actively choose what it wants to move in the direction it wants to move in. So even if I have 50 pieces of poison in my blood, my cell still wants to be able to dump the poison out into the blood. Right? So in order to move a substance against its concentration gradient, we use what's called active transport. You should recall from Bio 1 that active transport utilizes ATP as an energy source to push a molecule against its concentration gradient. Okay. Now, active transport always requires a protein facilitator. It can't just happen. And it is a very specific reaction. So this would be a protein that's geared very specifically to move triangles. Now, I have a low concentration of triangles here and a high concentration of triangles here, but my cell wants to move triangles out. Okay. The way that this will occur is that this region of this protein will have an affinity for this particular molecule. So when this molecule bumps into this domain, the protein will grab it and hold on to it. At this point in time, it still can't move it out because that would be against the concentration gradient. So it needs some kind of force to push it. And that force is supplied by this ATP. So once this is bound here, there'll be a conformation change here. Right? So this area is going to change shape, and it will be ready to receive an ATP. The ATP will fit into that region, and that last phosphate will break off of the ATP. This is now ADP, and it will float away to go regain another phosphate to become ATP again. And now this phosphate being stuck in this will change the shape of the protein again. And this time it's an energized change, right? The breaking of that bond released a little bit of energy to do some some free energy to do work. Right? And that work is going to be another change in shape of this protein. And this change in shape is going to force the triangle down and out. And so it's going to change shape and push the triangle out. And so that's how cells use ATP to move molecules. Right? It's all about changing shapes of proteins. So anytime a protein engages with another molecule that has a kind of chain reaction within the shape of the protein itself, and ultimately it changes its function. And in this case, those changes take the protein from grabbing to pushing. Active transport. So, onwards and upwards to osmosis. And now osmosis is specifically the movement of water in and out of a cell. And so, to start thinking about osmosis, we have to really talk about what the concentration of different molecules represents. So on the board, I have a diagram, it's a model. This dotted line represents a permeable barrier. Okay, so in this case, this would be the cell membrane when we're talking about actual cell transport. And so if we look at just the black dots, obviously there are more black dots on this side than there are on this side. However, the concentration of black dots is higher on this side. And so concentration isn't simply a measure of how much, in an absolute sense, there is of a solid on one side versus the other. It's a measure of how much of that substance there is compared to everything total. So on this side, I have 3 black dots and 1 green dot. So in total, I have 4 dots, 3 of which are black. So this is 75% black dots, or 3/4. Now, over here, I have 10 black dots, but I have 20 green dots. So overall, I have 30 dots, and only 10 of them are black. So over here, I have roughly 33%, 33.33% black dots. So my concentration is only 33%. So 75% versus 33%. So even though there's more black dots on this side, the black dots are going to move this way by the concentration gradient. Now in this situation, if this dotted line is permeable to all colors, the green are going to come this way, and then some black are going to come with them, right? And so I'll get a balance on each side. So eventually I should have a nice even distribution of black and green dots on all sides. That's only if this line is permeable to both substances. And now if it's permeable to both substances, we don't really need to talk about it in the storyline. When I'm talking about osmosis and the movement of water, the thing is, water is always going to be able to move across the membrane. Okay, the cell membrane has specific protein channels embedded in it that only move water. They're called aquaporins. Aqua meaning water, pore meaning hole. Right? So aquaporins. They're these holes that let water in and out. So water can always move across the membrane. Other substances, such as salt or sugar, can't as readily move across the membrane. Right? So in this model here, let us have the black dots represent water, and the green dots represent salt. Okay, salt being ionic, it can't just crisscross across the membrane like that. Okay. So because I have a higher concentration of water on this side than I do on this side, the black dots are going to move until I have equal concentration. And so I move one over here, and now I have 11 out of 30. Okay. And over here I still have 2/3, right? So 2/3 is still higher than 11 of 30. So I'll move another dot. And now I have 50% over here, and I have 12 out of 30. So it's still a lower concentration over here. So I'll move this over here. At which point now I have 0% concentration of water, and I have, what is it going to be, 13 and 30, whatever that math is. It's still less than half, right? At that point in time, the water can move back this way, but then it's just going to move right back, okay? Because these green dots can't move. So when I'm talking about osmosis, I'm talking about the movement of water, particularly in the context of concentration relative to substances that cannot move across the barrier. Right? Because the water can always move, these other substances can kind of manipulate the water. Be right back. So, when we're talking about osmosis, we're talking about it in the context of life. We're talking about it in the context of cells. And so we're comparing the amount of water by concentration inside the cell to outside the cell. And we're looking at how that difference in concentration makes the water move. So my circles represent my cells, and around the cells is the environment. I have three circumstances. I have a circumstance where the environment has a higher amount of stuff in it than inside the cell. I'm using salt as my example. So salt can't go in and out freely, but the water can. So hypertonic is when I have a higher stuff outside the cell. Okay? So therefore I have a lower water concentration outside of the cell. The prefix is referring to the stuff outside, right? So hyper means big, high, large, a lot, hyper, right? So I have a lot of stuff outside the cell. Isotonic means I have an equal concentration outside the cell, inside the cell, so there's no net movement. And hypotonic means I have less stuff outside the cell than inside the cell. So I have a higher water concentration inside than outside. So when we're talking about cells that have no stiff wall, these are very, very, very important ideas, right? So like inside of our cells. So if I am in a hypertonic solution, right, my blood has a lot of salt in it, or a lot of potassium, a lot of sugar, right? Anything that can't move across the membrane. Okay. And so therefore I have a lower water concentration outside than inside. The water is going to move out of my cell. It's trying to reach equilibrium with this solution out here. And so obviously as the water is moving out of the cell is going to start to shrivel, right? Because the water is what's keeping it nice and plump and healthy. So it loses the water, it shrivels. Okay? Isotonic is great. This is where we want to be. This keeps the cell nice and sexy and healthy. Hypotonic is the opposite of hypertonic. Right? And so I have a solution here, right? And the water outside is higher concentration than the water inside. So the water is going to rush into the cell, and that cell is going to swell. And if it gets bad enough, it's going to burst. Right? And so this is what happens if we don't have enough water or if we have too much other things. This is what happens if we drink too much water. You can die from drinking too much water. It happens. People do it to themselves every once in a while. It's really, really hard to drink that much water because usually you can just pee it out, sweat it out. Okay. But occasionally, every once in a while, somebody does kill themselves by drinking too much water. Best example I can think of is there was a woman who was preparing for a marathon, and so the day before she was just drinking and chugging and chugging and chugging and chugging, just gallon of water, just chugging it. And she got to the point where she drank so much water that she exploded her cells. So her internal organs began rupturing. She started bleeding from the inside out, and she died. Right? So that can happen. Don't want to do that. But here we are, hypertonic. I have a higher water concentration inside than outside. It rushes out. Hypotonic, I have a lower water concentration inside than out, so water rushes in. And then isotonic is equal and the same, balanced. So, when we are looking at the way in which water is going to move, we talk about it in terms of water potential. And so water potential is the sum of the pressure potential and the solute potential from one side to the next. So what that means, basically, is how hard are we pushing on the water and how much stuff is in the water. Tonicity. So if we have this fancy U-shaped tube set up, okay, I have a permeable barrier in the middle. It's permeable to water, but not the solid. In this case, I have 100% pure water and 100% pure water. Okay. So the solute potential is the same on each side. The pressure is 1 atmosphere coming down on this side and 1 atmosphere coming down on this side. So this is just an open air tube, right? So if you imagine we fill up a tube like this with water, right? And the water on both sides is going to be equal. Right? That's easy to imagine, right? Easy peasy. Now, pressure potential is really easy to picture, right? If I take a syringe thing and I push down on this side with more pressure, right? I push. If I push it this much down, then this side is going to raise that much up, right? That just makes sense. I push down the water, it has to go up. So I increase the pressure potential on this side. Okay, move it that way. That's changing pressure. I can also change the way this water is going to move by adding solute to one side. Okay. So I add some salt over here. Okay. And so on this side, I have now decreased my solute potential. Okay. So the water is going to move from my high concentration to the low concentration. It's actually going to be able to push up against gravity a little bit. So I'll drop on this side and I'll raise on that side. And the amount that this can go up is based on the concentration of solute that I add. The more salt that I add, the higher this can go. So it's going against the pressure of the atmosphere and the pull of gravity. Right? It's pushing against that pressure to go up. Now, I could also increase my pressure here. Right? So I take a syringe and I push back down, and I'll move the water back this way, and then there'd be this balance. So the amount of pressure that I'm putting on either side, combined with the amount of solid concentration that I have on either side, work together to form this water potential. It's deciding which way the water is going to move. Right? And so when I have a cell, right, it's based on if there's salt inside and out, or if there's a non-movable solute inside or out, right? But it's also based on the pressure of the cell. And so if this cell can squeeze itself, right, it can push stuff out. It might be able to push that water out. Okay? And so that comes into play when we're talking about things like plants, with those big central vacuoles and nice turgidness. Right? So water potential. Pressure potential, solid potential, combined equals water potential. Which way is the water going? Okay? So that's what we're looking at. So, what I'm going to talk about now is a little twist on the standard story of diffusion and gradients. And so we're still talking about going from high gradients to low gradients. The only thing is that now we are adding electrical charges. So I have ions that are involved in diffusion rather than neutral molecules. And so I made this diagram really, really, really simple. I put all of my cations up top and all of my anions on the bottom. Right? And so the cations are triangles and the anions are circles. So there is the concentration gradient here of the chemicals. Right? And so my triangles want to move down and my circles want to move up along their concentration gradients. That doesn't change at all. But I've also added these charges. So right now at the starting point, 1, 2, 3, 4, 5, I am plus 5 charge on the top, and I am 1, 2, 3, 4, 5, 6 minus 6 charge on the bottom. And so just like the concentration gradient for chemicals wants to equal out, the concentration gradient for charge wants to equal out as well. Right? And so the rate at which these move will be even faster than if it was just simple chemical gradients, right? And so now this diffusion is going to happen very, very, very fast because of the charge difference. So I don't just have the chemical gradient pushing this diffusion, I also have this electrical charge gradient pushing the diffusion. So instead of going whoop, it's going to go whoop, right? So the same diffusion, just happening faster because there's more at play. Okay? And so this electrochemical gradient is how our neurons fire so fast, right? And so we tend to think of our neurons as functioning on an electrical signal, which is a bit of a misnomer. Like it's not really like an electrical signal like the way that light bulbs work, right? It's working on this. There's a charge difference from one side of the membrane in the neuron to the other. And when we open the doors, they change really fast, which sends the signal really, really fast, which is great. Okay. So the electrochemical gradient is when you add charge to the story of diffusion. Okay. Just speeds things up a little bit. Can make things a little bit more complicated too. So like this was a really simplified version. All my pluses were one molecule, all my negatives were another. But for instance, I could have a couple of negative molecules up here, let's say squares, right, are negatives. Right? And so chemically, these squares want to move down by their concentration gradient, right? But electrically, there's a bit of resistance because this is already negative, and so they're like, I don't want to go to the negative space. And so they'll find a balance there. And so if down here is wicked negative, and the concentration gradient is only a little bit in favor of moving the square down, then the squares will stay up. Right? But if it's only a little negative, and I have a ton of squares, the chemical gradient will come into play, right? And so there's a balance. Yeah. And so whatever stronger wins until they're equal, and then they just go by diffusion. Cool. So that wraps up our introduction to cell transport. We got passive transport, which is just different types of diffusion, where our molecules are moving from a high concentration to a low concentration down that concentration gradient. The higher the gradient, the bigger the difference from one side to the next, the faster the diffusion will occur. We have active transport, where the cell is pushing a molecule against its concentration gradient. That requires energy on the cell's part, as well as a protein facilitator to actually move the molecule. We have the story of osmosis, which is the movement of water, often driven by non-permeable solutes. So we looked at the story of water potential, where the pressure on the water plus the solute concentration of the water impacts which way the water will move. And we looked at how adding electrical charges to the gradient impacts the movement of those molecules. And so we can manipulate electrogradient chemicals, or electrochemical gradients, to expedite cellular communication and the diffusion of certain substances across the membrane. And yeah, that's it. That's how cells move stuff. So in class, we're going to look at some of the more specific or more nuanced mechanisms, such as coupled protein transport thingies, which are fun. The sodium-potassium pump, which is utilized in a lot of cellular mechanisms, which is a story of both coupled protein pumps and electrochemical gradients. So it's a good study. And that's it. So I hope this has been a good refresher on cell transport for you. Have a fantastic day. I will see you next time.